Hello, everyone. Welcome to the Creation Podcast, the show where we discuss the science that confirms Scripture. I'm your host, Trey, and I have with me today Dr. Jeff Tomkins, ICR's Director of Research and Geneticist. Thank you for being on the show, Dr. Tomkins.
Well, it's great to be here.
Awesome. Well, today we're going to talk about something that I know that you are particularly passionate about, and it's one that evolutionary scientists have kind of been talking about for a while now, too. The supposed similarity between humans and chimpanzees. Now, we could discuss a bunch of different facets about this topic, but let's start at the very beginning of where this kind of idea came from. Why is it important to an evolutionist for humans and chimpanzees to be related, if you will?
Well, Charles Darwin, promoted the idea that humans evolved from apes in the 1800s, and that caught on with the evolutionary community.
Right.
And they didn't know which ape we were supposedly most similar to or had evolved most recently from. It was basically a toss-up between gorillas, orangutans, or chimpanzees. Mm-hmm. Actually, a study was published in 2009 by evolutionists showing that humans shared more anatomical similarities with an orangutan than any of the other great apes. But through the era of DNA sequencing and DNA research, it was determined that chimpanzees were probably the closest in their similarity to humans. So genetically speaking, exactly. So things have kind of focused on chimps as a result of that.
Okay.
But really, each one of the great apes and humans, they have their own unique features that make them special and unique. And so it's really, when you look at things like similarity, anatomically speaking, we share some features that are somewhat similar with chimps, others with orangutans, others with gorillas. So there's no clear path of ancestry, really, when you look at the overall creature.
So are evolutionists saying that we split, the tree idea, that we split from some other ape ancestor, some millions of years ago?
Yeah, they claim that 3 to 6 million years ago, we split from a common ancestor we shared with chimps, not that we descended directly from chimps, but that we descended from this mythical ape-like ancestor that was probably chimp-like, according to the evolutionary story.
Okay. That makes sense. Thank you for that background. Okay, so that brings us to, that was all in the 1800s. Now that kind of brings us more to the modern era. I know that in the not so distant past, although it's more in the past than it was yesterday, scientists sequenced the chimpanzee genome, and they created this big old project called the Chimpanzee Genome Project, right? Why would they do this?
Well, it kind of really goes back to the 1970s and early 1980s when they were taking human DNA and mixing it up with chimpanzee DNA, and they were unzipping the DNA based on the chemistry and the type of solutions and experiments they were doing, and they were seeing how fast it would combine human and chimp DNA together.
Okay.
This is in the laboratory. And based on these studies compared to the controls, which was just chimp with chimp and then human with human, they determined that chimpanzee and human DNA was close to 98% similar based on those studies. Well, those were not actual DNA sequencing studies. They were based on a technology called reassociation kinetics. And so it was a very indirect way of determining similarity. But it ended up being a gold standard, if you will, that later projects tried to adhere to, basically using cherry-picking techniques later on. But whenever you read chimpanzee-human DNA similarity study papers, say between the years 2000 and 2010, they always make reference to those original studies like they have to live up to them.
It's like, if you don't meet this standard, then the paper's not even worth it. So don't publish it or anything like that.
Exactly. It was like I said, a gold standard that just somehow had to be met.
That's interesting and kind of disheartening for the scientific community. You would think that if you did your study and you came up with a different number, that you would promote that number, even if it isn't the same as this past study, right? Okay. So the consensus of this project, you said 98% similar. Is that accurate? 98, 99, somewhere in there?
Yeah, the estimates basically, flow between 98 to 99. Well, when I say estimates, I say the gold standard paradigm. So typically you'll see 98.5, which is, halfway
in average,
in between. When I first came to ICR, I was asked to investigate the human-chimpanzee DNA similarity issue, and I had run a genome center at Clemson for about 5 years and did my postdoc in genomics, and I had never studied the human-chimpanzee DNA similarity issue.
Right.
And so I was asked to investigate it, and I said, okay, I don't know what I'll find, but when you start investigating something, you go to the literature and see what's been published already. So I began pulling up all these major papers in the scientific journals that claimed a 98.5 DNA similarity. Mm-hmm. And I was quickly discovering that these research projects were throwing out a lot of data that was non-similar, and they were cherry-picking regions of the human and chimpanzee genomes that were extremely similar. And in fact, some studies, I could actually factor the data back in to see what the real similarity was, and I was coming up with numbers anywhere between 66 and 86% similar, depending on the paper. So
that's a big difference from 98.
Yeah, at that point, I realized there was some monkey business going on with chimpanzee DNA research, comparing it to human.
Awesome. Okay. So you started studying this, and I'm just thinking, I mean, technology has just blown up in the last 50 years, right? And so in the 70s, I was not alive in the 70s. I don't know what the technology was like, but I imagine that especially in such like a budding science of like genomics and stuff, I imagine that the technology they were working with was not as sophisticated as what we have today.
Yeah, exactly. Well, DNA sequencing, it was invented in the 1970s, but it was very crude at that point and was just used for some very select regions, typically in microbial genomes. Okay. And then it improved in the 1980s with some new technology, and they were able to sequence larger pieces of genes, but they still weren't really sequencing genomes per se. And the genes they focused on were genes that were very common among mammals. So they were standard genes, or what we call housekeeping genes, that basically do basic physiological stuff in the cell. And they were very similar between humans and apes, and really humans and rabbits and other mammals. So. And then the Human Genome Project was conceived in the late 1980s and really picked up steam in the 1990s. And but the basic chemistry of the DNA sequencing was pretty similar to what it had originally been. It was called Sanger-style sequencing that had been invented in the 1970s. They just ramped it up with robotics and a technique called the polymerase chain reaction, and some other techniques. And so they just ramped up the DNA sequencing, but the chemistry stayed the same. And of course, the machines they used gradually, improved over time in their technology as well. But that's the basic chemistry that they eventually used to sequence the chimpanzee genome in the early 2000s, really between roughly about 2002 and 2010 was when the bulk of that project took place. And that took place with this Sanger-style sequencing, which had some problems as far as contamination related to it.
So not just technological issues, but actual contamination in the lab, I guess?
Exactly. Well, the technology itself was good, but the problem is when humans work in the lab, they have DNA on their fingertips and on their faces, and it's in the air, and you just have to be extremely careful about that DNA, that human DNA, getting into your samples. And we didn't really realize that this was a major problem during the 1990s and early part of the the 2000s era. So it turns out that several scientists published papers looking at databases for human DNA contamination. And they found out that it was a huge problem. There was human DNA in bacterial genome databases in various types of animals. In fact, they found that 3 million bases of a fish chromosome was entirely human. It was put together entirely human, and they actually published a paper on the genome with that mistake in it. Of course, they went back and fixed it later.
Right.
But this is a huge problem, and in fact, I did some studies with actual DNA sequence showing that indirectly that the chimpanzee genome was contaminated with human DNA in its early phases, and it probably went into the later phases. Now we have new technology now where we can avoid that contamination issue, and the chemistry, of course, is totally different with this new technology.
That's awesome. It sounds like we've made some major advances. So let's talk about your specific studies of this. So you were asked to when you came on board here at ICR to actually look into this, and so you found contamination. You found that there was just maybe some cherry picking there as well, and you did your own studies. What was your conclusion that you came to as far as the similarity between the chimpanzee and the human genome?
Actually, when I first began discovering that there's a lot of cherry picking going on in these research papers, I said, I need to just download my own DNA sequence and look at the data myself. And of course, I mentioned that I discovered, human DNA contamination, especially in the early first half of the chimp genome project, and it seemed to be less in the second half. And so when I began looking at those data sets that appeared to be less contaminated, I found out that the human and chimpanzee genomes were no more than 85% similar.
It's a big difference. Exactly,
using the standard algorithms that are out there that scientists use. Well, I also began downloading new DNA sequence data that was produced with brand new technologies. And these new DNA sequencing technologies use different chemistries than what I called the Sanger-style sequencing chemistry. It's a sequencing by synthesis type technology, and there's actually different chemistries that do that. But anyways, these chemistries produce really long sequences, which we call reads in the genomics business.
Okay.
They produce exceptionally long reads. The Sanger-style sequences produced about 700 to 1,000 bases per chunk, per read. And then there was a new technology after that called next generation technology, which produced actually very short reads, but it just produced more of them.
Okay.
And that technology produced reads that were 100 to 300 bases long when you would pair up the reads with each other. But this new technology can produce reads up to 10,000 to 50,000 bases in length, which are really long. And because it's sequencing by synthesis, you can pretty much tell if it's human or it's chimp because it's such a long read and because the technology kind of prohibits and alleviates the contamination issue. Okay. So anyways, they have now produced, having said all that, they have now produced a new chimpanzee genome based on this new technology, which is important because it's more accurate, it's less contaminated. And another thing is that the earlier chimpanzee genomes with the Sanger-style sequences, they actually assembled or put together those sequences based on the human genome. Mm. So the human genome was used as a scaffold to stitch together all of those little snippets of DNA that they produced that were only 700 to 1,000 bases long.
That seems like cheating to me.
Well, it is cheating, but they didn't have a whole lot of options because human was, the closest thing to chimp, and they didn't really have a super good framework, what we call genomic frameworks, for the chimpanzee genome, like they did other projects. And so they just used human to put it together. And another factor, too, was that there wasn't, a whole lot of money available for sequencing the chimp genome compared to, say, the human genome, where there's just billions upon billions of dollars that have been dumped into it.
Right.
Because we want to solve all of the health problems that humans have, and look at the genome in very much in depth. Getting back to the new technology that's being used to sequence the chimpanzee genome, I began downloading those huge snippets of DNA, and the data sets that I downloaded, on average, those little snippets were, on average, 30,000 bases long.
It's a lot longer than 100 to 300.
Exactly. It's great data to work with. So I began matching those up onto the human genome, and one of the problems I ran into is I could only match up about a third of those 30,000 base snippets onto the chimp genome. In other words, only about 10,000 bases of that I could actually match up before the algorithm just quit matching, because things became so dissimilar that it couldn't continue. But anyways, based on those 10,000 base on average snippets that I could match up, there was a maximum similarity of 84%, or an average similarity, of 84%. So meaning a maximum similarity of 84% between human and chimp. Well, that was pretty close to my other work that I did with the other DNA data set. So at this point, I would say that the human genome and the chimp genomes are no more than 84% similar. And that doesn't even include the regions that we can't match up, that haven't been sequenced yet, particularly in chimp.
And that is very different than 98, 99. And are there other, maybe like other secular scientists that are kind of hitting upon this same idea, maybe?
Yeah, exactly. That's a great question, because in the 2018 paper where they published the new version of the chimpanzee genome, using this new technology, they basically said previous versions of the chimpanzee genome were humanized. That was their words, humanized. Well, that was because they were assembled onto the human genome.
Right.
But they wouldn't commit to any similarity numbers between human and chimp, which I thought was odd.
That's very telling.
It was very telling, exactly. But there is a researcher in England, his name's Richard Buggs. He works for the University of London, Queen Mary. And he actually did an analysis similar to what I did, only he used a different algorithm. And he came up with the same number, 84% similarity between humans and chimpanzees. And actually, he published his research in a series of blogs that he put up on the internet, roughly at about the same time I published my data. So it was very providential that our results came out at the same time.
So you have someone who, because of course that's the way the scientific community is. It's, this creation scientist said this, so they'll just discount it. But someone else, a secular scientist, also said it at the same time. That is, providential is a very good word. Okay, so really, I want to bring it back to like the importance to the evolutionary community of like what this 84% really means to them. Because I imagine, 98, 99%, that's pretty close. Maybe we could have evolved in their worldview. But does 84% present a major problem to them?
Yeah, 84% is a huge problem because it kicks evolution out the door, basically. And the reason that is because we know what the mutation rates are in humans based on studies of large human families. And also within chimpanzee populations, we know what their mutation rate is amongst those populations of chimps. It's roughly similar to that of humans. So based on this idea of human evolution and the mutation rates that we know, and this model of human evolution called the neutral model of human evolution, and the statistical equations that go into all of that, you basically need about a 99% similarity to get your system to work. And that's within, say, a 3 to 6 million year time frame back to a common ancestor. Now, I've seen some papers where they try and increase the amount of time, up to, say, 16 million years, but even that doesn't help.
Yeah.
You basically need almost an identical DNA similarity between humans and chimps to get these evolutionary models to work that are very theoretical, but yet that's the hard fact. So 84% just totally blows that out the window. So really it speaks to the fact that humans are very unique. Their genome is unique. Yeah, we share similarities with other mammals and animals.
Because we breathe and have stomachs and
Right, because we have a lot of the same metabolism requirements, and even humans and chimps, have a somewhat similar body plan. It's not identical. There's obviously huge differences between them. And so you would expect a lot of similarities. You'd expect common code based on common biochemical and anatomical features.
That absolutely makes sense. So then I guess really what we're looking at is, if 84% is true, which it is, if we've got this 84% similarity, then that really just kind of pushes all of the evolutionary timescale back further than can work, right? 16 million, 6 million to 16 million in the grand scheme of the evolutionary timescale is not a lot, and I don't think that would account for a whole another 14% of the genome, right?
Right. And another interesting thing, too, is that the Y chromosomes of chimpanzees and humans were compared in excruciating detail about 10 years ago. And they actually created a very accurate map for the chimpanzee Y chromosome that they hadn't done for the other chromosomes. And so they knew that it was very, very accurate. And they compared it to human, and they compared the evolution of that chimpanzee chromosome to human to that of a human and a chicken. That's how different that was.
Wow.
Now, of course, the other regions of the genome are more similar, but
I think honestly it speaks to a common designer, right?
Exactly, right.
Well, and we know that we have been fearfully and wonderfully made. We are made in the image of God. We are not evolved from an ape-like ancestor. And all the glory to Christ Jesus for that. And you actually have a book on this topic that you wrote. It's a pretty recent book for our viewers. You can all see it there. Chimps and Humans, a geneticist discovers DNA evidence that challenges evolution. This came out not too long ago, if I'm correct.
Yeah, it just came out this past year. So I'm really excited about it. It has all the up-to-the-minute research that I've done, that secular scientists have done, and it's in a variety of different chapters on different topics. So we talk about DNA similarity in there, the chromosome fusion, junk DNA, pseudogenes, just the whole gamut of arguments that evolutionists have used to attempt to prove similarity between humans and chimps.
So it's a technical book, correct? Or pretty technical?
Well, it's a semi-technical book, and most people that have an interest in science and biology will be able to understand it at some level, some parts of it more than others.
Okay.
But I think it's pretty readable for the most part. Okay. And it's especially great for college students that are getting hit with evolutionary ideas in the classroom.
Absolutely.
And so I think it'll be great for a lot of different groups of people.
So to our listeners and viewers, of course, if you want to get more information on this particular topic, be sure to pick up a copy of this book. Again, that's Chimps and Humans. You can find that at icr.org/store. And for now, this is the first part of a 2-part episode. So make sure to come back and watch the next episode. So Dr. Tomkins, thank you so much for being here.
Thank you. It was great.
Absolutely. And for all of our listeners and viewers, again, this is just part 1. We'll have a part 2 on our next episode. And for now, make sure to like, subscribe, and share this episode with your friends. You can find it on YouTube or wherever you get your podcasts. And I'm Trey, and we'll see you next time on the Creation Podcast.